everyone with us today's webinar. In today's webinar, we investigate the dynamic field of tyrosine sulfations implications for synaptic plasticity and memory. So today we are honored to have Dr. Biswaranjan Sahoo as our guest speaker, as a postdoctoral fellow at the Dept Brain Research Institute in the Pharmacology, Physiology and Neuroscience Department, New Jersey Medical Schools, Newark. Sir, your expertise has significantly enhanced our webinar. The entire team of Heredity Biosciences greatly appreciated your willingness to share your knowledge with us. Thank you, sir, for your time and insightful presentation. But we are apologize for any delay for leading to this webinar and truly value your presence. The quote of this today's topic is, tyrosine sulfations play a crucial role in regulating protein interactions at synapses, influencing neuronal connectivity and adaptability. Its involvement in synaptic plasticity offers vital insight into memory formations and cognitive functions. So now I highlight to this today's topic is, so our guest of speaker will brief all these points, but I am highlighting some of the points of this. What is tyrosine sulfations? So tyrosine sulfation is a post-translational modification where a sulfate group is added to the hydroxyl group of the tyrosine amino acid residue. This process is critical for modeling protein functions. But the tyrosine sulfations, tyrosyl proteins, sulfotransferase, which are enzymes responsible for attending the sulfate group to tyrosine residue within the specific protein. So what is the biological importance is that the tyrosine sulfation play a key role in protein-protein interactions, enabling proper functions for protein involved in cellular communications and signaling. But it regulates binding and activities of receptors and ligands, including chemokine receptors, neurotransmitter receptors, and adenosine molecules, making it essential for maintaining healthy cellular processes. But this modification is particularly important in tissue like the brains, where the influence in neuronal signaling pathway related to the memory and synaptic plasticity. So now we'll move on the role of tyrosine sulfations in synaptic plasticity. So what is synaptic plasticity? That is, it refers to the ability to synapses that connected to between the neurons to strength to weaken over time to response increase or decrease in their activities. It is a fundamental process underlying learning and memory, where stronger synaptic connections lead to better informations, retention and processing. So second one is tyrosine sulfation's roles. So tyrosine sulfation's roles is to modulate receptor functions like chemokines and neurotransmitter receptors. Second is impact signaling pathway involved in synaptic strength and plasticity. Third one is evidence for studies showing how disrupted sulfations affected synaptic functions and plasticity. Continue for the highlight of this topic is tyrosine sulfation in memory. That is two types, that is implications in memory formation and potential therapeutic avenues. So tyrosine sulfations affect long-term potentials, a process vital for memory formations. Second one, alteration sulfations has been linked to cognitive defect in neuronal disorders. Continue on the potential therapeutic avenues, targeting sulfations pathway to treat memory-related disorders, that is Alzheimer's and cognitive declines. So final conclusion is that tyrosine sulfations play a crucial role in regulating synaptic plasticity and memory, making it a potential target for therapeutic interventions in cognitive disorders. So before I introduce you to this guest of speaker, let me say to the brief about this Heredity Biosciences. Heredity Biosciences is a leading company, is formed in 2018 and it's renovated in 2021, and Heredity Biosciences has growing into the prominent institution in the regions. It's involved into the pioneering research and development in the field of life sciences, contributing to new discoveries and technology that could revolutionize healthcare part, diagnosis part, and treatment, and some of the various research and disease diagnosis also. It is located in Mehfalagam Road, Joydeep, Bhubaneswar, and we have provided for the services in hospitals, license institute, and pharmaceutical companies. We have a wide range of applications like life sciences research, proteomics and metabolomic research, and genetic diagnosis and biomedical applications. So we have a long scientific advisory boards. They have a panel of think they are time to time guiding us to how to improvise to this content, how to serve to this better platform to this to the students, professionals also. So we are collaborating for this Heredity Life Sciences. Those are guiding us to and means participate to this this type of events to time dates. Heredity Biosciences have one of the portfolio that is a service platforms. So we have a wide range of service platforms. As you know that we have analytical services, metabolomic services, phytochemical services, microbial services, proteomic services, oligosynthesis services, artificial gene expression analysis, including gene expression analysis, real-time PCR services, and different gene expression analysis services, genomic services like identifications, bacterial identifications, fungal identification, plant identification, other identifications like other species identifications, as well as for the next generation sequencing, metagenomic sequencing, whole genome sequencing, and other part also. So all these services have a various part that are included in these slides. Heredity Biosciences have one another platform or another domains that is Heredity Bioacademy platforms. So these domain have this complete hands-on procedure of the students and the professionals. So those are lacking behind of this the hands-on exposure of this the experiments, so that we are providing for this in our laboratory. So that is included for this training and training, that is clinical microbiology, molecular biology, plant biotechnology, environmental biotechnology, food microbiology, aquaculture microbiology, pharmaceutical microbiology, some of the advanced modules that recombinant DNA technology, molecular cloning, molecular histopathology, proteomics and protein analysis, and some specialized modules have a wide applications to this laboratory like clinical embryology, real-time PCR, qPCR analysis for the gene expression analysis, advanced clinical genetics, and online modules of the bioinformatics also. Heredity Biosciences announcing for this dissertation master degree program. So those are interested for this program. So they are interested for these students. We have a wide applications for this dissertation programs for this product oriented as well as the research oriented topics, so that the students should be able to this and utilize to this for the future career. So these are the pictures of the witness of this last 1 month's have activities. So first websites we have to skill development program is ongoing. That is a free program of bioinstrumentations. Now we are going for this one of the free program, that is a biochemistry and chromatography of the programs. So more than of the students they are joined for these programs, and last 1 month we have completed for the 300 of the students. They are completed for this free program of bioinstrumentation as well as for this biochemistry as well and chromatography. The benefits of these students are a participant of the hands-on experience of the laboratories. And the right side part of this hands-on learning for the one day or 2 days program of this. So the students would be hands-on experiment of this molecular biology, microbiology, and other experimental things also that they have that enhance to the skills and laboratory skills, that understanding experimental techniques also. And our mentors, they are time to time guiding to expertise to how to utilize to this career to these experiments. As you know that we have some internship program is going on to that. So many researchers now have they are going for this real-time activities of this of the internship, that is clinical microbiology, molecular biology, environmental biotechnology, and other modules. This is the weakness of the students. Best of photos, and these are totally involved with this research oriented work, and they are involved into this for the future careers also. Another one domain of this hereditary biosciences are research activities. We have so many researches going on, and these are the researches like nanotechnology and microbiology and other part also. These are the things we are going on to that, and some of the paper we have to publish into the last 6 months. That is more than 30 publications and 3 patented we have to in our domain. Heredity Biosciences have focused to this, how do students have to join for these different organizations to that. As for that, till now we have more than of the 100 students are joined for the different industrial sector, research sector, and healthcare sectors also. These are the pictures to witness of these students. They are joined for the different organizations, and they join for the organizations. I wish you all these students the very best to this for the future career. Now, the time for this to introduce our guest of speaker. So we are honored to introduce Dr. Biswaranjan Sahoo. He is a distinguished neuroscientist currently serving as a postdoctoral fellow at the Newark Brain Health Institute within the Pharmacology, Physiology, and Neuroscience Department of New Jersey Medical Schools, USA. His academic journey is marked by notable achievements, including a PhD in neuroscience from this prestigious National Brain Research Institute, India. His doctoral research focused on the role of tyrosine sulfation in synaptic plasticity and memory, an area where he has made sustainable contributions. Throughout his career, Biswaranjan sir has gained expertise in a wide area of techniques central to neuroscience research, including epigeneticity, path climbing recording, in vitro electrophysiology, and molecular biology techniques. He has also trained internationally, acquiring advanced skills in electrophysiology and neuropathology through renowned programs in Germany and France. His research interest lies in understanding how molecular modifications, such as post-translational changes, affect synaptic functions and memory formations. His work on the tyrosine sulfation and its implications for synaptic plasticity has been published in prominent scientific journals such as Learning and Memory, and presenting at leading international conferences including Society for Neuroscience and EMU Synapse. In recognition of this innovative research, he has been awarded several prestigious level grants, including the Redgott Brain Health Institute Training Travel Award and the EMU Travel Grants. He has been involved in frontline research on social behavior in mice with an emphasis on how neuronal circuits adapt to environmental changes. He will be presenting today on this topic, tyrosine sulfations, implications for synaptic plasticity and memory, where he will investigate into the role of this critical post-translational modifications in the brain and its potential to influence cognitive health. His work offers important insight into neurological conditions and may lead to novel therapeutic strategies for cognitive disorders. Once again, thank you, sir, for joining us today. Please join me. Welcome to this Dr. Biswaranjan sir. So these are incredible team members. Those are time to time efforts to this webinar as well as to the training research part. Thank you. So now we are to join connected to take care of the section. This one.
Thank you so much, sir, for your kind introduction, and also I'm thankful to Sushree and Rashmi, and I'm so happy to join and to share my research work, and it's very special for me. And as I'm I started my bachelor's and master's from Bhubaneswar, and I am giving a talk to my city. It's my city people. I'm so happy to share my work. So, are you able to see the slides? Okay. Sorry for the delay. Okay. Thank you so much for inviting me. I'm very excited to share my research work, and I'm glad that the Heredity Biosciences actually and Dr. Purohit sir are doing a really great job. And okay, with that kind introduction, I'll go to going to share my story I have during my PhD, and if I have time permits, I'll go for the postdoc work. Okay, so the title of this talk is Tyrosine Sulfation Implication for Synaptic Plasticity and Memory, and Dr. Purohit already made easy for my thing the introduction. She has already introduced the topic to you guys. Okay, so human brain is the most complex piece of matter in the whole universe, as far as we know, and this is one of the most evolved structure, and it is what makes human being the most intelligent and the most advanced creature on earth. And our brain has different like the cortex, the cerebral cortex is divided in different lobes. For example, the frontal lobe, which is very important for planning and executive function, and the parietal lobe, which is there are all the sensory processes. For example, our touch and the taste, we that is processed here, and the occipital lobe is for the visual information. They're processed. Visual cortex is occipital lobe, and the temporal lobe is another most important lobe, housed the structures like hippocampus. Okay, I'll say hippocampus is one of the most important structures for memory formation, and like yes, so our brain consists of human brain almost 86 billions of neurons. Okay, and what makes different, like each person is consisting of this many neurons. Why we're different? Each person is different from others because the way the neurons are connected is different from each other. Okay, the experiences we get from the childhood till now is different from everyone, and these different ways of connections make and this is called synaptic connections, that makes us different from each other. And so why hippocampus? Why the name has been termed as hippocampus? Because see, in this photo, the left side, this is the dissected hippocampus from a human, and this is a seahorse. Okay, it's a marine fish whose scientific name is hippocampus. So the shape is, they are looking very similar. Okay, so that's why they have coined the term hippocampus, and it's very important for memory formation, as well as this is the GPS of our body. If someone asks what is the GPS in our body, because we are nowadays using Google Map. Okay, but when there was no Google Map, nothing, how we travel, how we find our location. So this is actually guiding it acts as like GPS. So all the behavior that we are any kind of behavior is the result of interaction of genes and environment. Okay, so and learning, so the process of acquiring new knowledge around the world is learning, and memory is when this knowledge we acquire is stored and we retrieve later. That is the memory. So based on how the memory lasts, how long it lasts, the storage, they divide into basically two forms. Okay, short-term memory, which lasts from seconds to minutes, and long-term memory, which lasts from hours to days to years. Okay, imagine an old man of 80 or 90 years. It also remembers from his childhood. Okay, so how the memory lasts from lifetime. And already you have introduced by Dr. Purohit, these post-session protein modifications, they actually play important role for this short-term memory. And long-term memory involves gene transcription and protein synthesis. Okay, so and this what happens during short-term memory? There are proteins, ion channels, which are majorly proteins, and when their conductivity increases, and neurons are very specialized cells where their performance, their excitability they're different from other cells in the way they have specialized ion channels which to carry out the functions. And during long-term memory, there is a growth of new synaptic connections. Okay, the proteins are synthesized, and there are growth of new synaptic connections. So I'll just share one of the. I want to show just one video. It's a short video. It's really important to. Are you able to see show my screen?
Yes, sir.
Yes.
Okay. So. So from different sensory information from different cortices, the visual cortex, the auditory cortex, and the sensory cortex, all the information, it comes, and the hippocampus, it's integrated all the information at the hippocampus. And once, the memory is formed, its long-term memory is formed, it again, the storage house are different at the different parts of the cortex. Okay, so the hippocampus memory formation start at the hippocampus, and again it goes back to different cortices for the storage. So we talk different languages, right? We talk Odia, we talk English, we talk in Hindi language. But how the neurons, in which language they talk? So they talk, their language is chemical. Usually their talk is chemical in form of neurotransmitters, and that happens during this synapse. So yeah, the two ways. Okay, so either here the existing synapses that is strengthened, the conductivity between these synapses increases, or there is a new formation of new synaptic growth. Okay. So within a neuron, the signal is transferred by the electrical, action potential. But between the neurons, it is a chemical in form of neurotransmitter. So the neuron preceding is presynaptic, and the neuron which receives the information is postsynaptic. Okay. So yeah, this is very important, see, very strong experience. For example, right now, if you are learning this new thing for you, then now in the hippocampus, the cells must be now firing, and they are, maybe some of the information are going to LTP is happening right now, the starting formation in your hippocampus right now. So with this introduction, again we'll go to the okay, are you able to see my slide?
Yes, sir.
Okay. So yeah, so you have already mentioned this in the video. The sensory experiences, all types of sensory information, from different cortices, it comes to the hippocampus, and then when the memory is formed, for initially it's either short-term memory, okay, and how this short-term memory are converted to long-term memory, okay. So this is the process is called memory consolidation, okay. So it can be two types. One is a system consolidation, which actually happens in the brain networks, okay, different structures within the brain, or it also or synaptic consolidation, okay, within a synapse. So the system consolidation, which happens from within the brain network, that can last very, very long. It can complete the process. It might take some months, some years, okay. But the synaptic consolidation, which happens within a single synapse or in between synapses, it can happen within minutes and hours. And this memory consolidation is believed to happen during sleep. So sleep plays an important role for converting our short-term memory to long-term memory. So it repeated, when the signal is very strong, or for example, we are studying for an exam, we are rehearsing, or we have a very strong experience. During that, the similar neural circuit is activated when it was being formed, okay. And what happens during sleep? The sleep has different phases: the rapid eye movement, REM, and non-REM. So it has been shown that during REM, this protein, like the protein molecules, the phosphorylation of, there is another protein called CREB, calcium response element binding protein, okay. When the CREB is phosphorylated, that has been shown that it plays an important role in memory formation. And what happens during sleep? In each day's activity, whenever we are doing any experience, we are learning something, so that has been replayed during sleep. It replays exactly the same way and by the repeated of replay, then finally the memories become strong. And there are different kinds of long-term memory. So there is a few memories which actually require conscious recall. For example, I'll ask one question to you. Who has the maximum number of centuries in cricket? You can say, or you can message, text in the chat box. Are you able to hear me?
Yes, sir.
Any students, who has the maximum number of centuries in cricket?
Sachin Tendulkar.
Okay. And one more question. So let's say I'll ask, who was the player of the match in the T20 World Cup final? Any answer?
Rohit Sharma.
In the T20 World Cup 2024. Students? T20 World Cup final 2024. India won. Virat Kohli. Okay, right. And so how do you know? Now we're exploring how do you know, like how do you say that Virat Kohli? Where the memory is remembered. So this kind of memory is called fact-based. Okay, we need your brain. You are trying to retrieve your information. Okay, so these are called semantic, the facts-based, and episodic is your personal experience. For example, you have been to Bhitarkanika National Park and you see the 23 feet crocodile. So that experience, that memory you have formed, that is your episodic, the personal experience. Okay. So these both memories are stored, and the helpful structures hippocampus and diencephalon, both thalamus and hypothalamus. Okay. And there are other forms of memory also, like which no need of conscious recalling. Okay. For example, you're cycling. Okay. When you're cycling, you don't remember okay, I have to recall. Okay. You just cycle by by it's a procedural memory skill you have learned. So initially it takes time, but once you learn skills, you don't need to remember anything. You automatically cycle, right? And the part of the brain involved is striatum, and cerebellum is also helpful for muscular. For example, you are swimming. Okay, swimming is another skill. So for this, cerebellum is needed, and for emotional and pure memory, another structure called amygdala. So these memories are not, you are not recalling consciously, but for fact or personal experience, you have to remember, like retrieve, consciously recall. So what happens when there is an injury to the brain areas responsible for your memory formation? So this is called amnesia. Okay, when either your memory is lost, the previous memory, which is called retrograde amnesia, or you cannot form new memories, called anterograde amnesia. Okay, so after the injury, there is no form. So the classic example is in the movie you have seen, Ghazni. Okay, so there he couldn't form, and after his brain was damaged, like he couldn't form new short-term memories. Okay, so he took case of anterograde amnesia. So I will share one few minute with this. He is one of the most famous patient in neuroscience called HM. His full name was Henry Molaison. So when he was at the age of 10, he was riding a cycle and he got an accident, and his brain was brain was stuck in the cycle. So after that, he developed a neurological condition called epileptic seizure. Okay, so which is very painful, and the doctor, they advised him at the age of 27, his part of the brain, they believed to where the pain was originating, the epileptic seizure, is the medial temporal lobe. Okay, as I first initially told, and this structure including hippocampus. So hippocampus is seated like in the temporal lobe. So one of the famous neurosurgeon, Wilder Penfield, they removed his brain parts. Okay, both the brain hemispheres. You see the normal brain. This is the HM's brain. Okay, and this is if you take the cross section at this plane, you'll get this normal brain, but this medial temporal structure has been lost. So after the removal of this surgery, the brain part, hippocampus, medial temporal lobe, yes, he was cured of epilepsy. There is no pain. But what led him, but devastating, was he couldn't form new long-term memories throughout his life. So when the doctor entered each time, he forgets. Every 50 minutes he forgets. Who are you? Like he asks. So the Brenda Milner, the person who studied for 30 years of HM, he has but the other form of memory was intact. Okay, he couldn't form new memory, and the last 1 or 2 years before the injury, he also forget. But the childhood memory was intact, the skillful memory was intact. So that means the different brain memory are formed in different brain areas. Okay, so already in the video you have been introduced LTP, which is cellular basis of memory. So this was discovered by Bliss and Lomo, and the two scientists in for Anderson's lab in Norway. And after that, the discovery, hippocampus was the most studied structure for LTP, long-term potentiation. And this is the brief circuit of hippocampus of a section of rat. So this is a very interesting structure, having the information flows from the trisynaptic pathway. It's a very famous pathway. So it's from the cortex the information goes, and this structure is called dentate gyrus because it looks like a teeth, okay, a little bit, and from there it goes to CA3, another structure. So dentate gyrus, CA3, and CA1, the three structure hippocampus. So the information flows from DG to dentate gyrus to CA3, from CA3 to CA1. Okay, so this pathway is called Moji fiber pathway, and this CA3 to CA1 called Safer collateral pathway. So my research focused on this Safer collateral pathway. So I'll be mostly my research focused on this pathway. And so little bit about the mechanism of LTP. What happens in the pre and postsynaptic cell when the LTP is induced? Okay, so there are two receptors in the glutamate neurotransmitter is in hippocampus. It's very abundant, and the two receptors it has, one is AMPA receptor, another is NMDA. So these receptors are when the neurotransmitter glutamate binds to the receptor, it directly opens the channels, and the ions, it's called the cation channels, the sodium ions, they flow to the presynaptic. This is the postsynaptic terminal. Okay, and after the neurons are depolarized. Depolarized means the positive current enters. Then there is a magnesium block happens in the NMDA receptor. Okay, so when more positive current flows inside the cell of postsynaptic neuron, then this positive, it repels the positive magnesium ion. Okay, then the NMDA is opened, and through NMDA receptor, this channel, the calcium ions along with sodium, they enter, and this calcium ions actually plays an important role for all the signaling, and we found in the long-term memory with thought of the protein synthesis and DNA transcription happen after this signal. Okay, so sir has already introduced, so I just skip this slide. So there are different types of post-transcription modification are helpful in learning and memory. The most important are phosphorylation and acetylation, but nothing was known about tyrosine sulfation about what is the role in memory. And sir has already introduced, but I'll just based on from where the sulfate group comes to the biological system. So there is a molecule called PAPS, phosphoadenyl phosphosulfate. This is the universal donor of sulfate group in biological system. Okay, so PAPS, when it is taken into the from the cytosol to the cell, inside the cell, intracellular, at the trans-Golgi network. The Golgi network has cis and trans, but so in this trans-Golgi network, the enzyme TPST, tyrosyl phosphorsulfotransferase. Okay, so this TPST enzyme actually catalyzes the reaction of tyrosine sulfation. So those in the blue dots, okay, those are present on this membrane of Golgi, trans-Golgi network. So they take the sulfate group from the PAPS, the biological, this molecule, and they donate to the receptor proteins. And you can see this small, brown part. This is the sulfate group added, and after the addition, either all the tyrosine sulfated proteins, either they are transmembrane or they are secreted to the outside the extracellular matrix. So this is an example that there are two isoforms of TPST enzyme. One is TPST1 and 2. So yeah, here is just an example, how this taking the PAPS and catalyzing these PAPS, this sulfate group added to the receptor protein. And the tyrosine sulfation, it has a really important role in brain development and neural maturation, and also outer segment of rod retina. They are also helpful when this this process. And there are a lot of abundant TPST expression in different brain regions. Okay, and TPST1 is usually higher. So directly going to the study. So the aim of the study was to investigate the role of tyrosine sulfation in LTP, okay, long-term potentiation, and to study the role of sulfation in LTM, long-term memory. The first part is in LTP. What is the role? So first question was whether hippocampal proteins are tyrosine sulfated or not. To check that, we took this is a rat brain. Okay, this is the medial temporal lobe of rat, and if you take out the hippocampus and section in a vibrotome, okay, just like a microtome, and you take the slices, and after thickness, after taking the hippocampus, you put in the because when you cut the section, they are under stress. So to keep them come to normal healthy condition, you keep some time, like you prepare it was incubated in artificial cerebrospinal fluid for around 2.5 hours. Then it was processed for the tissue was processed for STS and Western blotting, and it was probed with anti-sulfotyrosine antibody to check the level of protein sulfation, and we found that there are two bands at 105 and 120 kilodalton, which are actually tyrosine sulfated in hippocampus. So the next question was the inhibitor of using an inhibitor which is well known as sodium chlorate, and at what step this inhibits? The first step of PAPS synthesis. Okay, the PAPS synthase enzyme that was inhibited by the sodium chlorate, and there is no further process, sulfation group happens. So we 1 hour treatment of this sodium chlorate to the hippocampus slices, we found that there is significant reduction in the level of tyrosine sulfate in both bands. Okay, but in the upper band, the 120 kD was significantly lower than the lower band. Okay, so briefly I'll go what we studied next. Okay, so we studied the electrophysiology properties, the LTP. Okay, to check that, I briefly explain, we put it the procedure how we did it. Okay, LTP studies. So this is the real hippocampus we used for the LTP. So we use a tungsten electrode. So okay, simply, neurons can be stimulated by different. There are different ways for how the neurons can be stimulated. Okay, for example, they can be stimulated by electrical current. They can be used by mechanically. They can, when you touch the screen, okay, and the skin, then that is also the neurons are getting activated. It can be activated by light and chemical. So what I used here, I used electrical current, okay, to stimulate the neurons of the hippocampus, the CA3 fibers. Okay, the CA3 fibers, like CA3, these are the CA3 pyramidal neurons, and they project to CA1. Okay, I already explained CA3. This is the region, and CA1. So those fibers project to CA1. So they fibers connect, synaptic connection with the CA1 dendrites, CA1 pyramidal neuron dendrites. Okay, so I usually stimulate this safer collateral pathway. Okay, this pathway especially. This is CA3, this is CA1, and this is the fibers. So this I use a bipolar tungsten electrode and stimulate those fibers and record from CA1. Okay, this is CA1. Record from, and I was recording from extracellular. It's not intracellular. So when you record from extracellular, using a glass or pipette, okay, or silver electrode inside the glass pipette. So these are the process. The rat hippocampus, was sectioned to we call a slice, not section, because it's thick, little more thick, and it was incubated for 1 hour. Then we insert the electrode, okay, at the safer collateral pathway, and record from CA1. So here, first we decide how much current to use. Okay, so how do we decide? We first inject some current, then we check the response in voltage. Okay, especially the millivolt per millisecond, the slope. I'll explain the next slide, I think, how we measure. So then we found how much current will be used for each slice. Okay, we section the hippocampus and we take a slice. How do we decide? The decision point is making this curve, and you just increase the current where you found there is no more increase in the response. Okay, that's the maximum response. Okay, and then you stop there. Okay, and then how much current you use for your LTP stimulus when you induce the LTP. Okay, like now what we are going to do, first take the slice, use different current and stimulate this pathway, fiber pathway with current. So, and how to decide this? Let's say there is a maximum response. Okay, so we usually take 40% of the current of the maximum response. Okay, you will ask why 40%? Why not full? Because if you take full current, complete full current, maximum response, because we expect in the LTP, during LTP, the enhanced response we expect. So if you take completely 100%, there is no more increase. So that's why we keep around 40% of the maximum current. Okay, and then using different protocol, I'll explain my protocol in the next slide. Yes, so in the real, in the live hippocampus, like there are different the hippocampus pyramidal neurons, they fire differently, like the different frequency during a LTP. Okay, so people have like low frequency, which is very low hertz, 5 hertz, and high frequency is 100 hertz. So here I use a protocol using a high frequency stimulation, which is called a tetanus. So there are 4 tetanus was there. Each tetanus consists of 100 stimulation, So there are 5 tetanus, total 400 stimulation, and the in between 2 stimulation, 1 and 2, there is 5-minute interval. So 20 minutes the stimulation protocol was there, and each stimulation was for 100 here 100 stimulation, and total 400 stimulation. After 20 minutes, the protocol is we see the response. So this is little bit the screenshot I took when I was recording. So this is what you see in the computer, how. So you see whether there is a negative towards a downward response, So, but you see action potential always is always upward. If you have studied in have you studied, just checking, have you studied about action potential? Do you little bit know about action potential? Anybody? I want to yes or no?
Yes, sir.
Okay, good. Okay, good.
Yes, sir.
You always see action potential is always upward. So now you see there is a downward, right? So why it's downward? Because I was not recording from the inside the cell neuron. I was recording from outside in extracellular. So when the depolarization happens, okay, inside the neuron there is more sodium ions, all the cations flow. Then at that time, with compared to the cell, outside is negative, the current. So because usually the resting membrane potential is around 65 millivolt, and of the neuron usually. Okay, so when we during depolarization phase, when we're stimulating, the inside is more positive but outside is negative. So okay, so here 0, so there is less. That's why there is a downward, you see the downward curve, and you find any at any time point there is a slow, there is a value 0.1. Here is 0.17. So what is that 0.17? So this is the time axis in millisecond, and this is the voltage axis in millivolt. Okay, so here the slope between these two curves. Okay, the 2 dots, the black cursor you see. So actually the machine, the program, records this. What is the slope of this curve? Okay, so that is what in simple words, this is the voltage response. Okay, so we are injecting a certain current and getting the voltage response, and we are seeing whether that is increased or not after the stimulation. Okay, so next question was whether this SC, the sodium chloride, the inhibitor of sulfation, whether this is, if we inject, or we incubate the slices, whether this LTP, there is a reduction in the LTP or not, or impairment. So we found yes, the sodium chloride actually impairing this LTP. Okay, so you see this curve, these traces. Okay, so I'll just zoom out a little bit. So you see that I show you that cursor. Okay, so this is the before stimulation. Okay, and after LTP, like we use that protocol, so there is increased downward, more downward. That means more voltage, more slope is happening. So before and after, so there is a increase in the response, and this is the control, and this is the sodium chloride. You can see clearly there is a reduction in the response in the SC. Okay, the sodium chloride treated slices. Okay, so this is the protocol, like the method. Okay, so I told you first you take the slice and you incubate for 30 minutes. Then you started incubating in the sodium chloride solution. So we used 1 millimolar sodium chloride. Okay, for 1 hour, at least 1 hour we first incubate. Then so 30 minutes only like control with no treatment of SC. Okay, only artificial cerebrospinal fluid. Then next 1 hour is we incubated with sodium chloride 1 millimolar. After the 0 point, we started stimulating, the pressing the current started, the protocol, the LTP. So it will, the 4 burst I explained in the last slide, using that protocol, we stimulated the fibers. Okay, and the blue is the control, and the red is the sodium chloride treated. You see, just after the stimulation, there is highly increased in the response. Okay, so this response is I just forgot to mention. So these are called excitatory EPSP. Okay, excitatory post synaptic potential. Okay, So we already mentioned like the pre and post synaptic. All the responses here were recording from the post synaptic CA1, okay, fibers surrounding that, okay. So you see there is increase in the control, but in the SC there is reduction in the potential. So this is the baseline is percentage. So if the control baseline is 100%, how much increase after the stimulation? So you see at 20 minutes time, okay, we recorded, and we see the compared to the control was 160% increase LTP induction, but in the SC and the sodium chloride ones are less than 130. Okay, so there is a significant decrease, same as after 60 minutes. Okay, so after 60 minutes or still there is a LTP, means the potentiation is there in the control around 140, but in the sodium chloride, the inhibitor one, there is a reduction. So did you understand basically, the potentiation after the treatment of inhibitor, it got reduced. So is it clear, this part? I know it's a little complex. I tried to make it simple. Is it clear?
Yes, sir.
Okay.
Yes, sir.
Okay, great. Okay. So next, the question was, okay, the most interesting question we asked was whether if we increase this LTP stimulation, okay, whether that increases protein tyrosine sulfation or not. That was the question. And the similar protocol, but here the baseline recording was still 20 minutes recorded. Okay, control. The blue is, so the green is the control, and the blue is the stimulated, the LTP. Okay, so there are two groups of slices. Okay, one hippocampus slices, the control, they're not stimulated at all. Okay, they just incubated in the ACSF, artificial cerebrospinal fluid, for 45 minutes. Okay, but with the LTP induced ones, after 20 minutes of the baseline recording, okay, without stimulation, then you started inducing the LTP, okay, stimulated, and you find these are the responses. Okay, higher increase the responses. And to our the most interesting finding was, yes, after LTP stimulation, there is a significant increase in the protein tyrosine sulfation level. There are the two bands. Okay, the 120 kD as 105 kD. This is it is proved by anti-sulfotyrosine antibody in the Western world, and you see there is a significant increase in the protein tyrosine sulfation level after LTP stimulation. Okay, so we thought of, wow, so if LTP stimulation increases protein tyrosine sulfation, what are those proteins, okay, which are getting this tyrosine sulfated? We tried to find out the answer. So by using, have you all, I think hopefully you all know immunoprecipitation assay. Do you know? I think you are
Yes,
very
sir.
familiar.
Yes, sir.
Great. So using immunoprecipitation assay, I used the sulfotyrosine antibody to pull down those samples of hippocampus and probe with sulfotyrosine antibody, the same antibody, and we found that the lower band 105 kD. This is the length. I know because the antibody is not that great. So this is the lower band. It's getting pulled down. Okay, that means we put the antibody and it only specifically binds to those proteins which are getting tyrosine sulfated. So we found this band 105, but the upper band, in the immunoprecipitation, we never get that band. Okay, so it's always detect the lower band. So we thought of, okay, let's put this, we stain with silver staining, and we take this band, okay, 105 kD, and we send for liquid LCMS, okay, for identifying which proteins are there. Okay, so you are almost familiar with LCMS, liquid chromatography mass spectroscopy. Are you familiar?
Yes, sir. Yes,
Okay.
sir.
Okay.
Yes, sir.
Just simply, yeah. So in this process, the principle is individual components, are separated by ionization, and the separation of ions are based on mass by charge ratio. Okay, and there are a lot of the few proteins we got from the score, which are the target proteins, and having different scores. And what are these protein scores? So this mass calculates an overall protein score. Okay, so this number reflects the combined score of all observed the mass spectra that can be matched to an amino acid sequence within a protein. Okay, and we detected there is alpha-actinin 4, one protein having the highest score of protein score. So that means the probability of that protein to be alpha-actinin is very high. So we confirm that result using an sulfinator. There is a bioinformatics tool where that predict how many residues in the alpha-actinin 4 are tyrosine sulfated, and we found there are two residues at position 4 and 298 amino acid residues which have the probability of getting tyrosine sulfated. And what does this e-value suggest? The e-value suggests in a scale of 100, the lower the e-value, the higher the probability of that amino acid residues to be sulfated, tyrosine sulfated. And what we did, we reconfirmed. We use again the immunoprecipitation assay, and we tried to the same antibody we run, just like the hippocampus samples. And this time we same this is the 105 kD, the lower kD band, and we strip the antibody, sulfatase antibody, and probe the alpha-actinin antibody 4, alpha-actinin 4 antibody. So basically you strip that antibody and your proteins are there on the sample. So now the alpha-actinin 4, if the protein to be same, alpha-actinin 4, this antibody, this protein, then it should be the same position. You can find a band, and we can find at same position, we can find the there is a detection of alpha-actinin 4. Although these results are, I can say 80% is confirmed, but still the final it needs to be confirmed. So now let's come to the point what this alpha-actinin 4 is present in context of synaptic plasticity and memory. So this alpha-actinin 4 is present in the postsynaptic receptor density, which governs the glutamate receptor, another glutamate receptor called metabotropic glutamate receptor. We already talked about AMPA receptor and NMDA. So they are ionotropic, those directly binding opens the ion channels and the current flows. But this receptor, metabotropic receptors, those are signaling processed by the indirectly, through the G-coupled GPCR, G-coupled protein receptor, the signal mechanism. So this is the summary of part 1, the first part. Inhibition of sulfase impairs LTP, and tetraburst stimulus enhances protein tyrosine sulfation. And the LCMS indicates that the 105 kD protein to be alpha-actinin 4. So now I take a pause. I will ask the question answer session. First part, we can ask any question if you have, because before going to the second part. I need to understand whether you understood or any doubts, anything. You can ask the few questions for this first part now, rather than at the end. You may forget. Is there anybody any questions from this part?
Sir, can you again explain about LTP, long-term potentiation?
Okay, sure. So let me go. Okay, so what happens? So LTP, you saw from that video, right? The synapses, when there is a it's kind of use or lose it, just like a muscle, if you train in the gym or exercise, the muscle increases. Similarly, when there are certain synapses, for example, let's say the CA3 synapse to the CA1, when there is more activity. How the activity comes, for example, let's say you were studying for your exam. Tomorrow you have an exam, or let's say after 1 month you have an exam, you are studying. You first learn one subject, for example, now we are learning a new thing, So when you reread the things, then the synaptic, like the current, from this presynaptic to this postsynaptic neuron, that increases. Current increase means through this. I'll explain through this. So what happens? Like let's say you are, the current, the cell, the presynaptic cell, D is getting depolarized. That means the current from the outside, usually in the neuron, the extracellular neurons, okay, the extracellular space have more sodium ions than the intra. Okay, so during activation, when the neurons are this presynaptic neuron is getting activated by different stimulus. As I mentioned, the neurons can be stimulated by different ways. Okay, so they can be stimulated by electrically. They can be stimulated. Let's say I'm telling now, for example. Okay, now I'm telling my your auditory system receives the information through words. Okay, so that information is a stimulation. Okay, so that the neurons are getting stimulated in your auditory cortex, in the inner ear. Then finally the information is transported to the auditory cortex. So especially the sound. Okay, here the sound or the verb are the stimulation. The visual information can be, you are seeing now this picture, for example. Okay, this stimulation goes to your first through retina, it goes to your visual cortex, and the image is made there. So basically, neurons after stimulate, anyways, okay, current, electrical current, like chemically they stimulated, those neurons are from the resting, the more current is flowing inside the neuron. So they are getting more negative, depolarized. They call depolarization. So then that depolarization happens. So this neuron, presynaptic neuron, activating the postsynaptic neurons, and there what happens? There the current is flowing from the presynaptic to the extracellular matrix to the postsynaptic neuron. Okay, so when this strength, they're using more. They're talking. One neuron is talking to the other neuron in a very strongly. Then what happens? There is the strength, we call the strength, the connectivity is increases between the synapse. When this connectivity and this the postsynaptic neuron is activated for a long time after the stimulation, that is called LTP. Okay, so the connectivity between one synapse, it increases for a long time. Okay, and that is called long-term potentiation. This especially the activation of the postsynaptic neuron from the presynaptic, this increases this higher response increase for a long time. Okay, let's say for 1 hour, for 30 minutes, for 1 hour, for 3 hours, it lasts. That highest, the activated state, it lasts for few hours. Okay, so if it lasts more than 1 hour, it is called LTP, long-term potentiation. And this is the basis. This happening in synapses that is responsible for memory formation. That's why you remember, your memory is being formed. Is that answer? Did I answer your question? Did you understand?
Yes, sir.
A little bit more?
Yes,
Okay,
sir.
great. Any other question? Any other question? Any doubts in anything? I'll try to make it simple if I can. So you followed, right? Whatever, you followed the topic, the first part.
Yes, sir.
Okay. So if there is no question, I can move to the second part. But if you have any question, please ask me. I want everyone, at least something you should understand. Huh? Okay. So should I move to the second part, or if any no question, or if any question, you can ask me. You can stop me. Okay. So what do I understood? You understood or not? Okay. So if there is no response, I will consider you have understood the first part. So I'm moving to the second part. So that was in the cellular basis at the cell level, neuron level. Now at the behavior level, okay, the role of, we try to study the role of sulfation in long-term spatial memory. We thought of hippocampus is the GPS. I told you, right? This is the GPS. So they actually all the spatial memory, where you go. Let's say you're in the city, Bhubaneswar. From one city to your Heredity Biosciences, how do you travel? Let's say you are not using Google Map. Okay, your hippocampus is guiding to the building. Okay, so there is a very interesting task for to access memory in rats. So this is called a Morris water-maze task. Okay, so this is a huge tank full of water, and this is imaginarily divided into 4 quadrants. Quadrant 1, 2, 3, and 4. And the rats are trained, okay? The rats are released here, and we check what the rats are doing, okay? So we try to find out, to escape, because it doesn't like much into the water. Let's say it's so it try to escape from the swimming, okay? So there are a lot of the rat is trained for many trials, okay? And they're released from different quadrants, maybe from here, from here, from here. The different releasing points. We pseudo-randomly, 20 trials we train the rat. So what it does, initially when we release the rat, okay, I'll play the video maybe. Okay. So now I release the rat, okay, and there's a camera from above. It's tracking the rat, okay. And initially it doesn't know where the platform is, let's say, okay. I think it's a trained rat, I'll see. So it's now exploring where there is anything I can escape, okay, from. So it's searching and searching and searching, and finally it found the platform, okay. So once it found the platform, we let it, stay there for 30 seconds, okay. So during the 30 seconds, there are different cues are there. There is a doll here, there is a black box here, there is a rack here. So it tried to remember it remember the position of the platform with respect to these cues, okay. Like where my position is there, it is, that time the 30 seconds is crucial, okay. So the training is done. Now we check memory. How do you check memory? So like either we check short-term memory 1 hour after the training, or long-term memory 1 day, 24 hours after the training session ends, okay. So during long-term memory, what do we do? We remove the platform, okay. The platform was there. So sorry, the platform was there. We remove the platform from here, okay. Now release the rat and see what are the three parameters we seek for long-term memory. Is how long it takes to reach the platform. So let's say we release here. How long it takes, the time taken to reach the platform from the release site, that is called latency, okay. So the quicker the latency, less time it takes, better the memory. And the second is the fourth quadrant, okay. How much time spends in the fourth quadrant, okay, in respect to the total time, okay. Out of all the quadrants, how much time it spends in the fourth quadrant. And the another criteria is how much this annulus, okay, the previous platform, there is no platform now, we have already removed, okay. How long or how exactly it's precisely it's crossing this annulus, it's called a circular path. How many times it crosses this previous platform area, okay. So if it is a precise memory, it will cross this precise memory like the annulus, okay. So let's play the video, okay. So we release the rat. Now you are checking the long-term memory. So it directly goes to the platform. So it has a memory, okay. But it is not finding, okay. It's trying to search, where the platform is there. It was all here. So now it try to, maybe some other area it's going, and it's exploring, okay, basically, because it's not finding the platform. It was there, okay, already, but it's not finding now. So now it again is it's crossing. Now it crossed the annulus. So it has a very precise memory where the platform was there, okay. So then yes, so the more the number crosses, the better the memory we access, okay. So now we thought of, okay, using sodium chloride, the inhibitor of sulfation, if we inject IP in the interperitoneally, whether there is impairment in the memory or not, that we checked. And there are different doses we tried, and we found like 200 mg per kg body weight of rat had the maximum. This is the latency by the way, okay. So this is the latency time taken. So during training there is a less time, okay. Other training trials increases, they're taking less time to reach the platform. And during STM and LTM, see there is a significant the red one is the 200 mg, control is the green one, and there is a significant difference. They're taking more time in the sodium chloride treated animals, okay. So this is the procedure, the methods. We inject the 200 per mg per kg dose of sodium chlorate for 7 days, and also the control, the PBS also was injected. Okay, control animals versus treated. And on the 8th day after the injection, 1 hour after that, the same procedure I mentioned in the video, I was conducting, training and testing the short-term memory and long-term memory. So what we found was very interesting. This sodium chlorate, treated animals, they have impair in long-term spatial memory. How do you find that? See, so this is the latency time taken. So during the training, and the group blue is the control, and the red is the sodium chlorate treated animals. And you found this, there's a decrease in the latency as the training trial increases, which is good, which is now in both the groups. And there is no difference in the short-term memory. After 1 hour, they are actually taking similar time, both the groups, control and the inhibitors. But during LTM, there is a significant, the control, AC-treated control animals are taking more time than the control to reach the plateau. So there is a impairing memory. Same in the annulus crossing, okay, that previous platform area crossing, the controller have higher number of annulus crossing than the treated. You just note that there is no complete abolition of memory, but there is impairment in the memory. They're still crossing, right? So that means it's not completely abolishing, but there is an impairment. And this is the time taken in the platform quadrant. Also, there is a reduction in the fourth quadrant. They're spending less time than the control animals, those injected animals. And this is just a graph showing how they are crossing. This is the control. There are more crossing in the annulus crossing, but in the injected SC inhibitor one, there are only less crossing, and they are overall, they are not specific to fourth quadrant. And yes, the summary part 2. Part 2 is inhibition of sulfation impairs long-term memory without affecting learning and STM. Okay, it's not affecting this inhibitor, only affecting the long-term memory. So sulfation contributes to long-term spatial memory. So okay, so now the future prospects, so it will be very interesting to see the signaling pathways in the sulfation dependent inhibitor inhibition of LTP. How this inhibitor actually, which pathway it's acting, how it's impairing LTP and memory both, and study LTP and memory in especially this TPST1 and 2 enzyme. If you knock out those, how they're behaving on LTP. Okay, and to analyze the sulfation level, just like we did after the LTP induction. Similarly, we can study after memory training. Okay, you train them in the Morris Watermaze, then check their protein level. So, and this study was published in Learning and Memory. And okay, so what are the questions? Do you have time? Sir, do you have time more for the question answer? Because I thought if I have time, then I can little bit go for the postdoc work. But first, I love to take some questions from this part.
Sir, end of this webinar, we have 2 to 3 minutes of the time, so the audience would be asked the questions.
Okay. So sure. So can I go for little bit, not much, my postdoc work? So I'm thinking, they may forget because this story is different.
Yes, sir.
This work is different from. So I thought if you have any questions, I can take now. It's better.
Yes, sir.
So please, if any from the second part, long-term memory part, any questions, then please ask me. Any protocol, anything, please.
Hello, sir.
Yes.
So first of all, it was a very informative presentation. Sir, actually, I had a question that you use sodium chloride to see whether it impairs the long-term spatial memory.
Yes.
So why did you use that?
Oh, it's not sodium chloride. It's a sodium chlorate. I showed you. Yes, it is an inhibitor of sulfation. So I am using a sulfation inhibitor to check the memory impairment. It's not sodium chloride. Okay, it's sodium chloride inhibitor of sulfation.
Okay, sir. Yes, sir.
Yes. Any other questions? Otherwise, I'll ask definitely at the end session. I'll ask few questions if you understood or not. If you'll not ask me questions, I'll ask few questions at the end.
So, whether this condition plays a role in Alzheimer's or Parkinson's as well?
Okay.
So, the impairment.
That's a great question. So we don't know now. Okay. So nobody has tested, but since it is impairing the long-term memory, we definitely estimate. We can assume that that might be playing some role. So, but nobody has studied that. But we have to check in future. Like people have to see. But maybe you never know. There are different kinds of memories. There are different pathways. This is just one pathway and like one form of post modification. So, but yeah, you have to check whether it plays a role. But nobody has studied that. But that's a great question. Definitely, it would be very interesting to study this aspect. Okay, if it is impairing the long-term memory, if you check a Alzheimer model, first start with the rat model, you check whether there is impairment or not. Okay, yeah, that would be very interesting question, and it will be good to see that implication part. Nice question. Any other question?
Sir?
Yes.
Sir, is there any way to increase the tyrosine sulfation? Like if it improves the LTP or long-term memory, then is there any way we can sulfate the tyrosine present in the proteins like alpha-actinin?
Okay.
Induce more sulfation so that more memory can be improved if someone is facing.
So yeah, that's another point. You can see the other things. So we don't know. Let's say I'm just thinking the processes, all the physiological activities, which improves memory. For example, let's say exercise. For example, okay, people have been shown that I didn't. I've mentioned about that. So in the dentate gyrus, I showed the hippocampus structure, right? So CA3, the CA1 pathway, and the dentate gyrus, the first part, I told the teeth-like structure, okay? So there are a few brain areas. It has been found that when you exercise, there is enhancement of, that new neurons are formed in the brain. So since centuries, it was very, in the controversy whether our brain, human adult brain, is forming new neurons or not. Are we making new neurons or not? So still there are several studies very controversial, but still few group found every day we add around 50 neurons in one of the hippocampus, like left and right. Okay, so especially the dentate gyrus. So exercises increases this more like neurons, and those neurons when integrated in the dentate gyrus, okay, they're responsible for certain form of memory. So maybe this exercises or doing brain activity, like for example you brainstorming or thinking, or diet, that might for example the food, but these are the basic we to check the physiology in normally let's say the sulfation process. Okay, so there is a baseline level and there's a higher level, but yeah, you have to check that whether just by increasing sulfation level of few particular proteins, memory can improve or not. This is a question. This is a question you have to check, and we're not sure about that. It may not, it may or it may not also, right? Because just like LTP, it cannot go beyond a point. But if in case of Alzheimer or the memory disorder persons, it can be studied actually. That's a good way to diet. For example, diet and sleep, proper sleep. There are different ways how memory can be aging population can be controlled. For example, proper sleep, proper diet, exercises, healthy habits. There are a lot of things for you, all of us, not only aging population, for us all. These are the ways we can keep our brain healthy, and we can regular exercise is one of the most important for brain health, and it keeps our memory intact. It has been shown that regular exercises and proper diet, they play a major role in the memory formation. Any other questions?
No, sir.
Okay, so then I can briefly talk about my the work I am doing now in my postdoc here in Rutgers. So I'm interested, our lab is interested in social behavior, okay, and neuromodulation. How the neuromodulators like oxytocin hormones and vasopressin hormones, they are responsible for social interaction, and especially I'm interested how temperature influences, in social interaction in mice. Okay, so what we do, like we take two mice at a time, and at different temperature for 30 minutes we expose them. Okay, let's say cold temperature, which is around 15-16 degree, and room temperature, which is 22-25 degree, and warm temperature, which is around 34 to 37 degree. Okay, and we check how the interaction is there. Okay, and the huddle, actually in cold, in the wild, you have seen the penguins, especially. There are a lot of animals, including penguins, they actually huddle together to survive from the drastic cold. Okay, so that the reduces by cold, huddling together, a lot of huddle, like 100 animals, 50, 150 together, they close to each other, so that the less heat is released to the environment in the cold. So we thought of, okay, what happens in the mice? Okay, so we found that the huddling time, the body contact, are more in cold than in the warm temperature, and the blue are the males and the pink are the females. You see, the males huddle more than the females. Okay, we don't know why, we have to check, but the males little huddle more than the females, and between temperature there is a significant difference. Cold has a higher. Then we check there are few receptors. Okay, there are tiny receptors on our skin in the nerve terminals, which senses the cold and the warm temperature. Okay, so there are few receptors called TRPM8 and TRPM3. So TRPM8 is a receptor which is actually senses cold, and mostly classically it has been shown that it senses cold, but also few reports say that it also senses the warm temperature. And TRPM3, which is another warm sensor. Okay, and we found that if those knockout animals, okay, if you knock out these receptors from the animals, how they behave in the this direct social interaction test, and also the whole comparison between the So we found that this TRPM3, the warm sensor knockout, okay, the animals having known this TRPM3, they have a huddling more. Okay, they're not feeling warm. Okay, so they're huddling more than the control animals at all temperatures. And the TRPM8, which is a TRPM8, which is TRPM8 is a cold and both cold and warm sensor. They're in between the wild type control and the TRPM3, the warm sensor animals. Okay, and you see there is both males and females. So another interesting, I'll finish. This is the last slide. Okay, so we thought of, okay, if who is their choosing, their choice as a put towards them. Okay, there is a 3-chamber chamber. Okay, there's a short higher, okay, chamber, and barrier, a small barrier is there. So there are 3 chamber. Okay, so one side there is a mouse, a cage mate. Okay, just like a litter mate, your sibling. Okay, a cage mate mouse is there, and another there is a small object is there. Okay, and this is the temperature, cold, warm, and room temperature, and another temperature called thermoneutral temperature. Okay, which is the body doesn't need any more heat, okay, to maintain the body temperature. So which is the animal are very comfortable. And the first left side chamber, it was thermoneutral, which is around 29 degree. Okay, so we thought of, let's say in different temperatures, what is their choice, where they're going? They are going to the comfort zone. Okay, they are comfort at this temperature, 29 degree, or they're going to interact with the cage mate, their sibling, litter mate. Okay, so what we found, the wild type, actually during cold, so here the graph shows the positive, okay, 0 onwards positive. It says it's going towards the casemate, okay, and negative towards the thermoneutral, okay. This is comfort, this is casemate. So we found in the cold, they are not going to comfort zone, they are going to interact with the casemate because they're feeling cold. But unfortunately, the mouse is inside the case, inside a cup, okay, which they cannot huddle, but still it's going frequently visiting that mouse. Whereas the trypamate, the cold and warm sensor knockout, they don't have any preference, okay, towards that. So with that, yeah, I conclude my session, and this is the lab of my previous PhD at National Brain Research Center. And thank my supervisor, Professor Shiv Kumar Sharma, and all my lab mates. And currently, I am in Rutgers, so I have a group of lab. My current PI, Dr. Yona Garcia, and my whole lab mates. And I'm thankful and the funding resource of DBT Department of Biotechnology for my PhD study, and National Institute of Health for the reason. And this is the beautiful campus of National Brain Research Center, where I did my PhD. Thank you so much. Thank you for your attention. And I love to take questions.
Thank you, sir, for nice presentation. So, the students, any of questions? The open for the questions over. Now you ask the questions.
Hello?
Yes.
Hello. Sir, can you tell me the significance of studying the social thermoregulation which you are doing like?
Okay, great. So, yes, what is the significance of this? So, you know what? There in this global warming is happening, okay? So, there is the behavior changes, okay? Few examples I'll tell human beings, okay? So, it has been studies shows that during warm temperature, when the climate is warm, okay, the crime rate increases in different, crime rate increases, the aggressive behavior of people increases, and they're thinking the brain also, though there is a change in the brain structure, health of the brain, this impaired. So we need to understand how in cold, also cold is a major stress behavior, okay? When in the animals in the cold situation, they're under stress. So their behavior changes also. They to combat the stress, cold stress, they go to huddle each other to maintain their body temperature, as well the behavior wise also, right? So it's very important to know what is the brain structure. For example, it's known that hypothalamus, okay, which is the major brain structure responsible for this thermoregulation, temperature regulation, as well as the oxytocin hormones in the PV and paraventricular nucleus of the hypothalamus. They also regulates this affiliative behaviors, okay? So it's important to which brain structure, how the neural circuit, which neurons are responsible for that. So if we understand in the global, especially the global warming case, and the brain behavior and the brain health is changing, so we need to understand how the brain structure is changing, at what molecule level, at the synaptic levels, at the brain levels, so that we'll be able to some preventive measures we can take. We understand what can be done about that. So it's very important. There are a lot of studies now going on because the impact of global warming on the brain health and the brain, and people are finding very surprising results. The way our brain behaves completely drastically changes during warm temperature, and as well as cold. But now in context of global warming, it's very important to know, and so that when there is a impairment or behavior, we can think of the how we can implications in the disease conditions or in the impairment conditions, and also studying the mechanism. How one small example I'll give you. This I told you there are 4 different temperatures. This thermoneutral zone I told you at 29 degree for mouse. Okay, so people have shown the 2 groups. Okay, animals. One was in the room temperature. Actually, room temperature is a little cold. Room temperature is 22, 25. It is actually cold for mouse or rats, and especially mouse. It is cold. They're feeling there is little cold stress. But if we put them in 29 degree, but we all people who are doing research in mice, we keeping them at the room temperature, which is 22 degree, which is cold. They're stressed actually. Okay, but one group study they showed that the mouse are under the room temperature and the mouse in 29 degree. They found nothing they have done. They're telling the growth of cancer progression was much slower in the 29 degree. They found nothing they did, no extra, all keeping all conditions same. They found the growth of because during this 29 degree thermoneutral temperature, cancer, for example, it needs a lot of energy. All the energy of the normal functioning cells, they are taking, the cancer cell is utilizing. So the body needs a lot of energy expending unnecessarily. So when at this 29 degree, the body doesn't need any extra energy to maintain your temperature. So they're saving a lot of energy. So that energy is utilized by the normal function. So they found that cancer growth and progression is very less hampered in 29 degrees. Just simply changing the temperature to 29 degree, it changes a lot. And in human also they found the range is a little wider, not like 29 in mouse. In human, so that our thermoneutral zone lies around. It's a broad range actually, from 24 to 35 something. It's a broad range, Yeah, but Now we see the basic mechanism is studying in animals, and we think of what implications found in the human being. But there are many human studies on this thermoregulation part. Most temperature regulation as well as social, both ways. Does it answer a little bit to your question?
Yes, sir. Yes, sir.
Okay. Any other questions? Am I beyond time? Sorry. Okay. If any question, I would love to take. If there are no question, up to.
So one question my side.
Yes, sir.
So what are the key career paths for the research opportunities for the students? Those are participating here and interested for the starting of this topic, that is synaptic plasticity and memory. So particularly focusing to the post-transcription modifications. What are your suggestions for this? The students would be career for this.
Oh, okay. So the students are like now what they're doing. They're master students or the PhD. What level they are now?
Basically master degrees students.
Okay.
All are the master degree students.
Okay. So so neuroscience. When I start in a PhD in neuroscience, they can from any background they can come to neuroscience, because neuroscience is a very interdisciplinary study. So I have seen my friends from different backgrounds. Like I am from geology, but I have seen people from biotechnology, mathematics, chemistry, physics. They all come because there is a computational, brain is so complex to understand that we need help of all other. So, to study, if you want to do neuroscience, we have now. I'm seeing. See, my interest is to become a scientist, neuroscientist, established scientist, but. Using this neuroscience, you can use go to industry. There are a lot of in industry also. There are now a lot of positions for helping this neuroscience people. They can in the industry as well as especially the artificial intelligence field. You can being a neuroscientist, you can go and help together, working with them, different models. And for you like bioscience, you can do a PhD degree in any Indian institute or as well as abroad, by applications like Europe or US. You can do a PhD in neuroscience from your background. And like any specific should I explain, sir? What exactly you want to know?
So basically all these subject oriented students, they will be focused for this experimental things, and they will be joined for this neuroscience program also.
Yeah. So it's excellent actually. Because when I was in my masters, I didn't get much scope doing hands-on. So actually you are in a good place. I'll tell that at least you have now hands-on training. So when you apply for PhD position in abroad, for example, they need some research experience. So you have done this internship, so it will help you. They will basically look positions for what skills you have there.
Yes.
But for PhD, they need little, if you have skills, really better, because if you want at least few experience in already hands-on research training, then they will really your profile will be very good. So they are the likely, but you have to know the procedure, So there are a lot of applications happens in Europe. For example, Max Planck. You have to check. You have actually there is LinkedIn is a good source for looking at the PhD positions. And when we thought, 12 years back, it was not, we didn't know much about. Now, check LinkedIn, there are a lot of positions open. You can go which field you choose, what are the applications, when the date is there. So you just have to keep updates from different university in different countries. And the process is like usually abroad, you have to apply, in the application, there are two things. One is through a program up there, in let's say neuroscience program or biology program up there, and through your just emailing to the PI. Let's say you are interested in some of the check institute, there are good labs, you find their work very interesting, so you email the PI, okay, the personal investigator email, and if you providing your CV, your experience, you write a cover letter like 1 page, okay, you write them, and you found either their position or they are also interested. So then you email and you think, okay, I found this position or I found your work, and you cite few of their recent publication, so that this is work I very interesting, and just few lines what you want to do in this lab, okay, in 1 page you just write that, and I would love to join your team to pursue my research career. I'll be very enthusiastic. You can email them, and this is one way. And the open program position, you have to apply their program through your CV, your all the transcripts, the certificates, and then you have to apply them, and you called for interview, okay. Then in the interview they will ask your basically your experience in your masters, any internship, any research prior experience. Then based on that they will select you.
So basically need for this hands-on experience, so accordingly they will be selected for this series.
Yeah, this is really important. Like if you have more hands-on training, they will hire those people who are already skilled. So but still if you write a good proposal, let's say plan, that is also sometimes very helpful. You even if you will not work in that particular project, but then just need to know how much you think about your project, your research plan. Okay, you have this plan, you want to do this. And so how you think, basically they'll assess your thought, thinking ability to put a question, a research question. Sometimes that also helps. Yes. And if anybody needs any help, if I can help, please write to me. I can provide my email to Dr. Purohit sir and also Sushree. Yeah. If anything I can help.
I will share to your mail ID to all these students also, sir. So if
Yes.
there will be any need for these questions or any need for the help, so they will directly mail you.
Sure. And thank you so much for inviting me, for giving me a chance to interact with students and to present my talk. And I'm very happy being a local, from my city, Bhubaneswar. It's very special. And I'm so happy for the Heredity Biosciences. They're doing really good job. Because during my time, we didn't have much hands-on training, and now at least in my city, the institute is providing internship to people, so it's students. So it's really, I'm so happy, and thank you so much. You're doing this work and this institute.
Thank you, sir, for accepting our invitations also, and all this audience, all the students also, they are waiting for this last 22 to 25 days. So they're eagerly waiting for that. So many students there join for this program. And any of these questions for these student sides, or else we will wind up these sessions.
But I hope you all make some long-term memories today. Your hippocampus will be.
This is a very good presentation, and I also learn a lot, because this is new topic for me also. But because I am in a reproductive biology and genomics also. So that's why it's new for me also. And
Yeah, it was also
students-
challenging for me. I know the students are from different background, and I don't know how much I can explain because it's a neuroscience little different. Like people usually molecular biology and all they're all study, but I don't know much how they studied. So it was really challenging for me. I tried my best to make it simple. I don't know how much. Yeah, you could convey.
Okay, sir. Okay, audience, the students will be end of this session. So first, lastly, I have lot of thanks to this Biswaranjan sirs. So finally, on behalf of Heredity Biosciences, I would like to extend my heartfelt gratitude to all of the part of this today's session. It has been a great insight and enriching experience today. And with sir, first and foremost, I want to express my deepest thanks to Biswaranjan sir for this enlightening presentation on tyrosine sulfation, implications of simplicity, plasticity, and memory. Sir, your deepest knowledge and ability to explain such a complex topic, really I love it, has truly inspired all the students. I also inspired through that. And we appreciate the time and efforts you have put into the sharing your research experience with us. Your insight into this role of tyrosine sulfations is cognitive health for great potential to the future development in neurosciences and therapeutic strategies. And all these students also, they will be learn a lot, and they will be utilizing to his or her careers also. And I would like to acknowledge to this Miss Sushree Swagatika, also coordinated to you, sir, and for her efforts, a lot of efforts behind this behind the scenes. So encouraging that every details to attach to that, and every details to coordinate it to you also. Finally, a big thanks to all of the participant attendees, your engagement and active participants, and encourage the questions also, and this events more valuable. We hope we will leave to today. I'm not detached to this presentation, because this is a lot of knowledge I gathered today, and with this knowledge and perspective I will gather here today. With that, I conclude today's sessions, and I again heartfelt thanks to Biswaranjan sir for your presence and wonderful presentation, sir. And I look forward and seeing all of these future events, collaborations, as well as for any of these experimental events also we could collaborate with you. And have a great evening, sir. And thank you, sir. Thank you for accepting again. Again, I put accepting for our invitations. Thank you, sir. Thank you, and have a wonderful day. Thank you, sir.
Thank you so much, sir, for your kind words, and thank you everyone for joining. It was a great pleasure to research, share my research work with you all. Thank you so much. Have a good night.
Thank you, sir.
Thank you, sir.
Thank you. Bye bye.
So finally, the students, we already shared to the feedback link in your message box.